today I'll be discussing on the nutritional requirement and the growth media. Under this, we'll see the various nutrients that are required by microbes for their growth characteristics, and the various types of growth media and the means of media sterilization. So, substances that are used in biosynthesis and energy production that are required for microbial growth are termed as nutrients. So these microbial cells is composed of over 95% of few elements, that is carbon, oxygen, hydrogen, nitrogen, sulfur, phosphorus, potassium, calcium, magnesium and iron. Such are termed as macroelements or macronutrients because they are relatively required by the microorganism in a very large amount, while all microorganisms also required microelements or micronutrients which are required in a very small amounts, and these are known as trace elements. So these trace elements include manganese, zinc, cobalt, molybdenum, nickel and copper. So now we'll come to the macronutrients, which we also know it as the macroelements. So elements that are required in large amounts, that is carbon, oxygen, hydrogen, nitrogen, sulfur and phosphorus, are components of carbohydrates, lipids, proteins and nucleic acid. So other macronutrients, that is potassium, calcium, magnesium and iron, they exist in the cells as cations. So this potassium is required for the activity by a number of enzymes, including those that are involved in protein synthesis. And this calcium, it contributes to the heat resistance of bacterial endospore. Further, magnesium serves as a cofactor for many enzyme complexes with ATP, and it stabilizes ribosome and cell membrane. Finally, iron is a part of cytochrome and a cofactor for enzymes and electron-carrying proteins. So next, we'll see these micronutrients or the trace elements. These trace elements are normally a part of enzyme and cofactors, and they aid in the catalysis of reactions and maintenance of protein structure. So, when we talk about zinc, it is present at the active site of some of the enzymes. Manganese aids in many enzymes catalyzing the transfer of phosphate groups. Further, in case of molybdenum, this is required in nitrogen fixation, while cobalt is a component of vitamin B12. Next, we'll see the requirements for carbon, hydrogen and oxygen. So, carbon is needed for the skeleton or backbone of all the organic molecules, and molecules serving as the carbon source normally also contributes both oxygen and hydrogen atoms. So, carbon dioxide is one of the important carbon source that does not supply hydrogen or energy. And microbes that use carbon dioxide as the sole source of carbon are the autotrophs, whereas those microorganisms that uses reduced preformed organic molecules as the carbon source are termed as heterotrophs. And the nutritional requirements of microorganisms, they vary enormously among different species. Also, nutritional requirement within a species may change because of mutations. A microorganism requiring the same nutrients as most of the naturally occurring member of the species are termed as prototrophs. Mutation may occur in this prototrophic organism, and hence it cannot further synthesize a molecule which is essential for growth and reproduction. Hence, the nutrients must therefore be obtained from the surrounding. So such microbes are termed as auxotrophs. Now we'll come to the nutritional types of microorganisms. So microbes are grouped into nutritional classes, and this is based on their requirement of their sources of energy, hydrogen and electrons. So based on the sources of energy, they are classified as phototrophs and chemotrophs. Phototrophic organisms use light as their energy source, while chemotrophs obtain energy from the oxidation of chemical compounds, which may be either organic or inorganic. Further, microorganisms based on their sources of hydrogen atom or electrons, they are classified as lithotrophs and the organotrophs. So organisms that use these reduced inorganic substances as their electron source are the lithotrophs, whereas organotrophs extract the electrons or the hydrogen from the organic compounds. And further, these microorganisms based on their primary source of energy, hydrogen or electrons and carbon, they may be placed in one of the four nutritional classes. So the first one is the photolithotrophic autotrophs. So these organisms use light energy and carbon dioxide as the carbon source, and most of them they employ water as an electron donor and they release oxygen. One of the examples is the algae, purple and green sulfur bacteria, which we also call it as cyanobacteria. So the second one is photoorganotrophic heterotrophs. So these organisms use light energy and they depend on organic compounds for their source of electrons and carbon. For example, purple non-sulfur bacteria and green non-sulfur bacteria. The third one is chemolithotrophic autotrophs. So organisms under this group use inorganic compounds for their source of energy and electrons and uses carbon dioxide as their carbon source. Organisms come under this include the sulfur oxidizing bacteria, hydrogen bacteria, nitrifying bacteria and the iron bacteria. So the last one is the chemoorganotrophic heterotrophs. These chemoorganotrophic heterotrophs use these organic compounds for their chemical energy source, electron and the carbon source. For example, protozoa, fungi and most of the non-photosynthetic bacteria comes under this group. Next we'll see the growth characteristics of bacteria. So we all know that the ultimate goal of any species is to make more of its own kind. So this fundamental law ensures survival of the species. So an increase in cellular constituent is termed as growth, and a typical bacterium grows by increasing its length and mass, which facilitates the expansion of its nucleate as its DNA replicates. So as this DNA replication comes near to completion, the cell, in response to the complex genetic signal, it begins to synthesize an equatorial septum that ultimately separates the two daughter cells. And such process is known as binary fission. In this process, that is the binary fission, one parent cell will split into two daughter cells. So usually microorganisms are cultivated in the liquid medium and they are grown in the batch culture or closed system. And as no fresh medium is provided during incubation, nutrient concentration declines and the concentration of the waste increases. Further, the growth of the microorganism can be plotted as the logarithm of the cell number versus the incubation time. And the resulting curve, it has 4 distinct phases. First one is the lag phase. The second one is the log or exponential phase. The third one is the stationary phase. And finally, the fourth one is the death or the decline phase. First, we'll see this lag phase. So during this period or during the lag phase, there is no immediate increase in cell number or mass when microbes are introduced into the fresh medium. And therefore, this period is called the lag phase. And during the lag phase, the cell, it synthesizes new components, although cell division does not take place. And this phase varies considerably in length with the condition of the microorganism and the nature of the medium. This phase, that is the lag phase, may be quite long if the inoculum is from an old culture or one that has been refrigerated. So next we'll come to the exponential phase. So microorganism grows and divides at the maximal rate during this phase, that is the exponential or the log phase. Their rate of growth is constant during the exponential phase, that is, the microorganisms are dividing and doubling in number at regular interval. As each individual divides at slightly different moment, the growth curve rises smoothly. And the population of the microorganism is considered to be most uniform in terms of chemical and physiological properties during this phase. And hence, these exponential phase culture are usually used in biochemical and physiological studies. Now we'll see the stationary phase. So microbial population growth ceases and the growth curve becomes horizontal during a phase that is known as the stationary phase. During this phase, that is a stationary phase, the bacteria attain a population level of around 10 to the power 9 cells per ml. But the final population size depends on the nutrient availability and the other factor such as the type of microorganism being cultured. So during this phase, the total number of viable microorganisms remains constant. This may result from a balance between cell division and cell death, or the population may simply cease to divide, though they remain metabolically active. And this microbial population enter the stationary phase due to several factors. The first factor is the nutrient limitation or nutrient deprivation. The second one is limited oxygen availability. And the last one is the accumulation of toxic waste product. Now we'll come to the death phase. So various detrimental environmental changes such as nutrient deprivation and the accumulation of toxic waste, it leads to the decline in the number of viable cells. So such a phase is termed as the decline phase. So the death of a microbial population is like its growth during the exponential phase, which is usually logarithmic. That is, a constant proportion of cells dies every hour. Although most of the microbial population dies logarithmically, the death rate may decrease after the population has been drastically reduced. And this is because of the extended survival period of a particular resistant cell. So now we'll come to the growth media of bacteria. So culture media is defined as any material that is prepared for the growth of bacteria in a laboratory. Bacteria can be grown in synthetic or defined media. The medium in which all components are known is known as the synthetic or the defined media. For example, many chemoorganotrophic heterotrophs are grown in defined media with glucose as the carbon source and ammonium salt as the nitrogen source. So these defined media are widely used for research purposes, as it is desirable to know what the experimental microorganism is metabolizing. So the second media is the complex media. Media that contain some ingredient of unknown chemical composition is termed as the complex media. And such media are very useful because this single complex medium is sufficiently rich to meet the nutritional requirements of many microbes. So this complex media contains undefined constituents like peptones, meat extract and yeast extract. So now we'll come to the different types of media. The first one is the selective media. Selective media favors the growth of one organism over another. For example, bile salt or dyes like basic fusion and crystal violet favors the growth of gram-negative bacteria, thereby inhibiting the gram-positive bacteria. And the second one is the differential media. These differential media distinguish between different groups of bacteria. For example, the blood agar, McConkey agar, etc. These are the differential media. So, blood agar distinguishes between the hemolytic and the non-hemolytic bacteria. While McConkey agar, it contains lactose and the neutral red dye that differentiate the lactose fermenting colonies that appears pink to red in color from the non-lactose fermenters. So next we'll come to media sterilization. So the process by which all living cells, viable spores, viruses and virions are either destroyed or removed from the object or habitat is termed as sterilization. So first we'll see the physical method of media sterilization. So heat sterilization is usually employed for the sterilization of media, and the most widely used method of heat sterilization is the moist heat sterilization, which is carried out at a temperature above 100 degrees Celsius in order to destroy the bacterial endospores. And this is achieved by the use of saturated steam under pressure. So steam sterilization is carried out by the use of a device which is known as the autoclave. In this device, that is autoclave, water is boiled to produce steam, which is then released through the jacket and to the autoclave chamber. So the air that is present already in the chamber is forced out until the chamber is filled with saturated steam and the outlets are closed. Further, hot steam, it continues to enter the chamber until it reaches the desired temperature and pressure, which is usually 121 degrees Celsius at 15 pounds of pressure. So at this temperature, the saturated steam, it destroys all the vegetative cells and the endospores. So this treatment is continued for 15 minutes to provide a margin of safety. So moist heat, it effectively kills microbes by degrading its nucleic acid and by denaturing enzymes and other essential proteins. So now I'll conclude today's discussion by stating that microorganisms require certain essential macro and micronutrients for their growth. Microorganisms can be placed in one of a few nutritional categories on the basis of their requirements for carbon, energy, and hydrogen atoms or electrons. Culture media provides all the necessary requirements for the growth of microbes, and these media must have to be free from all living cells, viable spores, viruses, and viroids. To achieve this, sterilization of media is carried out, and one of the most widely used method is the moist heat sterilization by the use of steam under pressure.